And for the last 3.5 years I've been working here at Ag Research in the Reproductive Technologies group, working on epigenetics and understanding reprogramming during the process of cloning of cattle. For about 10 or 15 years now, people have been sequencing whole genomes. It started with the Human Genome Project quite a number of years ago. And back when people started sequencing the human genome, we thought that that would give us all the information that we would need to understand development and complex diseases and everything we ever needed to know in science, or at least biology. But it's turned out that that's not the case. We have the complete sequences of quite a number of different genomes now, but we're finding that we still have many of the same questions. And for some time now, it's been known that this is due to something called epigenetics, which is an additional layer of information on top of the genome sequence. And over the last 5 or 10 years, this has really grown exponentially, this field, as people try and work out how the genome is interpreted in the context of the cell or organism. So epigenetics really refers to any sort of modification to the genome. It doesn't change the genome sequence, but it modifies the way that genome is interpreted by the cell. And there are different types of epigenetics. There are things such as DNA methylation, there are modifications to the histones, there are microRNAs, and all sorts of different ways that the genome can be packaged differently. So for many years, people have known about DNA methylation for many, many years, but it's been notoriously hard to measure, up until probably the last 5 years, where there's just been an explosion of different techniques on how to measure DNA methylation. When we clone, we need to take an adult cell that has its own epigenetic signature, and we put it into an egg cell which has had its DNA removed. But the epigenetic signature of the adult cell needs to be completely stripped off, and the cell needs to reapply the epigenetic signature of an embryonic cell, so that it can become a 1-cell or 2-cell, 4-cell embryo, and then develop through to being born and adulthood. This is obviously a hugely complex task because the mammalian genome has about 3 billion base pairs, and so it's amazing that we actually get any cloned animals at all, given what has to happen. If we can measure, just take a couple of cells from embryos that we've generated in the lab and analyse to see how well they've reprogrammed at least their DNA methylation, we can select those embryos that have much greater chance of making it. So if we can select the 20 best embryos and put them into 20 cows and still end up with 10 embryos, that saves us a lot of time and money and a lot of effort, because a lot of effort goes into looking after these cows when they're pregnant and calving them.